So our last speaker is Simon Bullock from the LMB in Cambridge. And of course, he was David's postdoc at the LRI from 1999 to 2004. And Simon has done a huge amount of organization for this wonderful day. So the title of his talk is Pair Rules for Recognition of Localizing mRNA. Over to you.
Great, thank you, Caroline. Yes, it's really actually quite difficult for me to put into words just how much David has meant to me throughout my career in terms of his mentoring and his friendship. I would, of course, endorse everything that we've heard about today and in the discussions in the breaks about David's remarkable scientific insights, his generosity, his infectious enthusiasm. I think in the absence of being able to find exactly the right words, what I'm going to do today is to try and pay tribute to David by sharing some recent unpublished work which actually provides some answers to a question that we started to address together when I joined David's lab remarkably about 25 years ago. So I'm going to focus mostly on the science, and I will say a little bit more about David at the end. Okay, so the story starts here in the blastoderm embryo of Drosophila. So Trudy and Alana have already introduced this system. So as you heard, at this stage of development, a subset of RNAs, such as those parallel transcripts shown in green and red, are transported apically of the nuclei in which they're transcribed by the Dynein motor. And I worked on various aspects of this system with David, but the big idea, the first idea we had, was to try to define what features of these particular RNAs and other RNAs that use this system in other tissues in the fly use to interact with the localization machinery. And that turned out to be much more difficult than we'd anticipated. And that's because when we look at the localization signals, so these are the regions of the RNA which are necessary and sufficient for dynein-based trafficking. When we look at them, although they all make stem loops, they have nothing obvious in common in primary sequence or secondary structure. And that led some groups at the time to suggest that there must be different RNA binding proteins which bind each of these and link them to the motor complex. And others suggested that, well, maybe you have a bunch of RNA binding proteins and they get together in some combinatorial way to provide the linkage. And then shortly after I moved to Cambridge, actually we showed there's a single RNA binding protein, which is called egalitarian or EGAL, which Ruth Lehman's lab had worked on previously, which contacts all of those RNA localization signals I showed you. And we also showed that egalitarian binds another protein called big D, which Eric and Ruth Stewart and others had characterized and turned out to be an important adapter between dynein and its cargo. So this explains how these RNAs are linked to the motor for microtubule-based transport. And that was satisfying, but it gave us another puzzle. And that's because when we looked at the sequence of egalitarian, it lacks a canonical double-stranded RNA binding domain. So we still didn't understand how these different RNAs were singled out for transport. And then this really was a structural problem, right? And the difficulty we face is that at this stage, the structural methods needed to solve structures of egalitarian bound to different RNAs just weren't sufficiently developed. So we moved on and we worked on for many years on various other aspects of microtubule-based transport. And then a few years ago, inspired partly by David's continued enthusiasm for this question and advances in cryo-EM, we decided we'd have another crack at this, and we collaborated with a superb structural biologist in Andrew Carter's lab at the LMB called Kashish Singh. Kashish worked closely with Mark and Sabella in my lab to try to tackle this again. And this slide shows you the first result from this collaboration, which is a cryo-EM structure of the egalitarian big D complex bound to a localization signal from a transcript called K10. So that localization signal is called a TLS. And what this structure showed us is that 2 copies of egalitarian recruit 2 copies of the localization signal to the big D molecule. And one of the most remarkable things about this structure was that each of these localization signals is actually contacted by the two different egalitarian polypeptides within the complex. And we have some really nice single molecule data that I don't have time to show you that shows that in this mechanism, this localization signal stabilizes this egalitarian dimer, and that's really the key step in allowing dynein to start walking along the microtubule when it's bound to a cargo. If we look more closely, we can see that egalitarian uses 4 non-canonical RNA binding domains to bind the localization signals. So there's this ED1 domain, it's the lotus domain, which contacts the minor groove at the bottom of the stem. There's a structurally very similar domain, which we call ED2, which does the same at the upper part of the stem in the minor groove. There's a domain called the XAD. I won't go into why we call it that. which contacts the major groove at the top of the localization signal. And then there's an exonuclease homology domain, which wraps around and contacts the other side of the stem loop. And it really appears that the RNA is scaffolding the assembly of these protein complexes, because the interfaces between the different protein domains is not sufficient really that they'd be stable in isolation. So the RNA is playing an active role in scaffolding this structure. But what then about the other localization signals? How does the egalitarian bind those? And we half expected that the protein would engage these different RNAs, which, remember, we couldn't see anything obvious in similarity between them, used in different combinations of these RNA binding modules. And we thought about this because egalitarian has a third low-cis domain, which we call ED3, which does not bind TLS and wasn't present in our structure. However, when Kashu solved a lot more structures of different localization signals bound to egalitarian, we found that they all bind the RNA in a remarkably similar way. So using ED1, ED2, the XAD, and this exonuclease homology domain. And that's because these RNAs turn out to actually share quite a lot of similarities with one another that become evident when we can compare their 3D structures, but were not obvious from the secondary structure predictions. So the first of those shared features is a bend in the helical axis. So this is a superimposition of all the localization signals I showed you. And they all have a bend, so they have a remarkably similar backbone shape. And that's essential for binding egalitarian because a straight helix, like this canonical double-stranded RNA, simply wouldn't fit in that egalitarian RNA binding pocket. And this bending is to do with these 3' bulges, and which basically allows this kink to form. And we have in vivo evidence that this is important for localization because if we delete these bulges from the localization signals in all those transcripts I described, so K10, hairy, the I factor, bicoid, and gherkin, we strongly disrupt apical localization when we inject the RNA into the embryo. And then the second shared feature is there's a pattern of base pair identities at precisely spaced apart positions in the upper and lower helices. So these are the secondary structures of the empirically determined RNAs, which are actually different from the thermodynamic secondary structure predictions we'd all been working for many years previously. And when we look at these structures, we can see that at this position, there's always a UA base pair, and here there's mostly a UA base pair, but sometimes it's a CU. So in the case of bicoid, there's a non-canonical CU base pair. And this can be explained by a really interesting mechanism. So this ED1 site, these side chains are inserted into the minor groove, and this position of this cysteine, which actually was a null mutation found in Drosophila genetic screens. This cysteine would cause a clash if there was a G base in this position. And at ED2, these residues insert into the minor groove and coordinate an iron or a water, which would similarly cause a clash with a guanine. So in other words, egalitarian is not making specific patterns of hydrogen bonds to recognize specific bases. It's the absence of a guanine at either the 3' or the 5' position that enables high affinity binding of the protein. And again, we can support this idea in vivo by mutating these UA base pairs at different positions to GCs in the TLS, either individually or together, and that strongly disrupts localization. So it's still unclear why we often see UAs, because anything that doesn't have a G should work, but I can speculate about that later if anyone's interested. And then the third layer of specificity was hinted at actually in a paper I published with David in 2003, and I also remember not being able to say surprisingly or importantly. But what we showed in this paper is for the hairy transcript, if you look at the 3' UTR, there are two distinct localization elements. There's two stem loop structures, and I don't have time to show you the data, but what we've gone on to show is for at least hairy and K10, and we suspect this is more general, the localizing RNAs need two egalitarian binding structures in a coincidence detection mechanism, and these egalitarian binding structures are contacting the different RNA binding interfaces of that egalitarian big D complex. And that's what I've said, and we can see that actually because we've solved the structure of one of these bipartite signals from hairy, so the work we did with David in this paper, bound to egalitarian and big D. And this second stem loop structure which we call a support element, still has the kink and it still has the base pairs lacking the guanine at specific positions. So it binds ED1 and ED2, but it can't bind the other RNA binding modules. And this means it's low affinity binding relative to the primary element. And this is also a feature we've seen in other examples of this for other transcripts. So we think this is selected for, and we think this is selected for because it allows reversibility to the interaction. So once the RNA is delivered apically, egalitarian has a probability of dissociating, so the transport machinery can be redeployed for other rounds of transport. So just 2 more slides, and I'm going to tell you briefly some take-home measures to say a bit more about David. So I've told you first that egalitarian recognizes RNA localization signals with a combination of non-canonical double-stranded RNA binding domains, that there are multiple layers that govern specific recognition of the RNA elements in the absence of obvious conservation of sequence or secondary structure. The first of these is recognition of a kinked RNA backbone. The second is sequence discrimination using this unusual guanine exclusion mechanism at distant sites, 2 base pairs that are always precisely spaced apart. And the third is detection of 2 egalitarian binding elements on the same transcripts. And together we think this adds up to give very highly selective transport of specific transcripts. And we think that this mechanism or similar mechanisms could contribute to recognition in other systems, not just to do with RNA trafficking, in which there's a RNA binding protein or a set of RNA binding proteins that target RNAs that have structured elements that seem to be diverse, and we would argue that maybe they do have elusive things in common. Okay, and then finally, obviously I want to finish by, I mentioned the people who did the work, and I want to finish by thanking David for so much, really. I mean, he set me off on this project, which we've worked on and off for the last 25 years. He was extremely generous to allow me to take most of this away with me, which is amazing. And also, as many people in the audience have been in David's lab will attest, you know, he mentors you in the lab, but he's a lifelong mentor, so he will always be there if you need support and give you advice. So I really appreciate that. And I have to say, I did have the great pleasure of taking David through the key aspects I presented today when he last visited me in Cambridge, and it brought a big smile to his face. So I hope you liked the story too, but if you didn't, I don't really mind because David liked it. And that's a good enough seal of approval for me. So thanks very much.
Thank you very much, Simon. Anyone have any questions? So, sorry. Nick.
So those different elements, the different ED elements, are they very tightly linked in the structure, or is there some flexibility that would allow them to kind of assemble themselves around the?
Yeah, it's a good question. Let's go back quickly. So, go back to the previous slide. Oh, sorry, thank you. Yeah, so there's a linker in the primary sequence between these 2 domains, which means that actually you have this mechanism where the ED1 and ED2 can't make this interface on the same polypeptide, so they have to do this domain swapping. So this link is quite short, the ED3 linker is quite large, but we haven't seen ED3 binding to anything, even though it's actually very conserved. So there might be some RNA targets in flies that we haven't worked with yet which use that. So in principle, you could span quite a lot of space, I think.
And Jippy had a question.
Thank you, Simon. This conversation and this kind of problem brought me back a few years, and the conversation that we had with David in the corridor. So what do you think that is the evolutionary pressure that made this beautiful mechanism switch in higher organism? Why EGL function has been replaced?
Yeah, I mean, we don't know, obviously, the sequence of things. So egalitarian, as GP implies, is specific to invertebrates, and we don't know what's the ancestral form. So actually, we have some data in the lab that says in mammals, the equivalent proteins to egalitarian are FXR1 and FXR2, which have similar features. They interact with big D through a similar region. They obviously have different RNA binding targets, and it may be actually that egalitarian as it evolved, you know, that's the kind of derived situation in a way, maybe because of things like oogenesis, where there's selective pressure to transport RNAs, you know, from one site to another over tens of microns. So I think in mammals, it might turn out that the FXR proteins are equivalent, but lots of the mechanisms that we found by studying the system actually seem to apply to the mammalian system as well.
Great. So we will finish the session here. Thanks, Simon, and the other three speakers as well for fantastic talks, and it's been wonderful.